Today we're going to talk about neuroanatomy, the study of the structure of the brain. Of course, we can't talk about the relationship between structure and function without first understanding what those structures are. And so think of today's lecture as a kind of roadmap, a kind of guide of where in the brain are we talking about when we're talking about the superior temporal gyrus. So maybe the challenge of one of today's lecture is that there will be a lot of Latinate terms and they sound impressive, but logically they're actually quite simple, right? It's simply a map of the brain and the central nervous system. Now, I like to start talking about this map from the bottom up, right? So I'm going to start by structural features of the neurons, right? So individual neurons and what they look like. And then I'm going to connect them together to talk about neural circuits. And then I'm going to start from the other direction, talk about overall, general overarching divisions of the nervous system. And then I'll go into the nitty-gritty. I'll talk about gray matter, structure, anatomy, and then I'll talk about fluid-filled ventricles, which are fluid-filled spaces in the brain and central nervous system, and the arteries, the blood supply, the vasculature of the central nervous system, and then we'll talk about white matter structure, right? So that's the outline of today. Let's start by talking about the structural features of neurons. Now, I know you've all heard about neurons, right? I'm pretty sure that you wouldn't have taken this course if you hadn't heard about what a neuron is. Neurons are cells of the nervous system, right? We have 10 to the 12 of them. Nobody has, it's just an estimate. Nobody's actually counted. But everybody who's taken one neuroscience or even psychology course has some idea of what a neuron looks like or does. Okay, so let's do a little pop quiz. Draw me a neuron, right? Take a piece of paper and draw what you think a neuron looks like. I'm just going to let you play me again when you're done. Okay, when you're done, I'm going to show you what most undergraduate drawings look like. And then I'm going to show you what postgraduates and postdocs drawings look like and lab directors and what their drawings look like. Now, if you just look at a group of undergrads, now this is actually published work on neuroscience education that was done by Hay et al. In 2013. What they've done was they've asked undergrads, postgrads, and postdocs and lab directors of neuroscience labs to draw them neurons. Now, if you look at what 7 of these undergraduates are drawing, the neurons look remarkably similar, right? There's always a nice cell body. There's a nucleus, right, in all of these cells, and then there's a cell body, and it has some dendrites coming towards it, and then it has this nice long axon, and then it's got these axon terminals, and so you can see that there's remarkable similarity between the undergrads' drawings. Now, then you can ask the postgrads, right, the grad students and the postdocs, what they think of, and you start to see this divergence between different individuals' conceptualizations, or how they think about what a neuron looks like. And you start to see that some people think of it as a cartoonish thing with some nice white matter bundles around them. Other people think of them as more sort of tadpole-like structures with a little bit more of a sprout at the top, right? And then when you get to the lab directors, what I find quite interesting is that they're really quite schematized, right? So this one professor just thinks about a neuron as 2 inputs and 1 output, right? Just a triangle. And then this one is much more of I guess a cytologist, right? Somebody who looks into microscopes and looks at all the dendritic tree structure. Now, but I think what's quite interesting is that undergrads have a very prototypical representation of neurons. And so what exactly do I mean by prototype? Well, when we talk about neurons, let's say in NSNB 213 or in Psych 105, you've heard about the basic features of brain cells or neurons. They all have some dendrites, right, and that go into the cell body, and then they go in through an axon into an axon terminal, right? And we know that the dendrites are what incorporate the input of information from other neurons into this particular cell, and then this cell then passes on the information that it gets from the dendrite down the myelin sheath, right, which is wrapped around the axon bundle, right? So the axon is this long part of the neuron, and so the information comes down the axon towards the axon terminal, right? So dendrites, axons, axon terminals. Undergraduate education starts with introducing these basic features of neurons, right? And then around the axon, you also have individual cells that are wrapped around the axon. These are called Schwann cells, which together make up a myelin sheath, right? And so that the purposes of this myelin sheath is to help the electrical transmission of action potentials that go down the length of the axon towards the axon terminal, right? But when you think about how the prototype actually translates into the 10 to the 12 neurons that you have, you start, in reality, there are actually many basic neuron types, right? So there are some neurons, such as the bipolar neurons that are in the retina. They can have quite a bipolar distribution, right, of the dendrites and axons. And then you can also have unipolar unipolar neurons that just have a single structure, a single length down between the dendrites and the axons, right? When you're talking about the prototypical neuron, you're mostly talking about the multipolar neurons that have multiple dendrites that go into the cell body, which then go down the axon towards the axon terminal. But certainly we have other kinds of dendritic arborizations or tree structures that characterize different kinds of cells. So a pyramidal cell, for instance, has this very dramatic tree-like structure. And this is another example of dendrites. So dendrites just mean a tree structure. And so when you have these tree-like structures that are connected to the cell body, right? That's how the cell receives input, right, through the dendrites. And here's another example of a retinal bipolar neuron. It's simply a cell body with dendrites on one end and axons on the other. Again, this is a cortical pyramidal cell, right, that is in the cortical layer, compared to retinal bipolar neuron, which has a much simpler relationship between the dendrite and the cell body and the axon. The cortical pyramidal neuron has much more of these dendritic arborizations, right? So it really branches out a lot more. And then when you get into the cerebellum, there's another class of neurons called Purkinje cells that really show a much more dramatic arborization or tree-like structure, and that's throughout the cross-section of the cerebellum. Now, in addition to neurons, we also have glial cells, and these are glia is Greek for glue. And so glial cells are just the glue-like cells. So they aren't really neurons, but they're helper cells that are serving as the glue for the neurons themselves, right? And so there are a few different kinds of glial cells. One of those is astrocytes. And astrocytes, the purpose of astrocytes is to provide nutrients towards the neurons that they wrap around. We've already talked about Schwann cells, right? So Schwann cells are the small cells that form the myelin sheath, which wrap around the axons to enable rapid transmission of action potentials. But we also have microglial cells. So these are just smaller cells that again serve as a cleanup or supply nutrients towards the neurons. And then we also have ependymal cells and also oligodendrocytes, and these are other kinds of cells that provide electrical insulation for the axons in the neurons themselves. Now, so I'm not going to play this little animation, but I do encourage you to click on it and the published set of slides and just look through the animation for how a chemical synapse happens. And I'll just suffice it to say that neurons work together by synapsing, by forming synapses into each other. And so that's the relationship between two neurons. There's a presynaptic and a postsynaptic neuron, and together these combinations of pre and postsynaptic neurons form larger neural circuits. Right? So we'll move on to talk about neural circuits now. Now, neural circuits, of course, is a circuit is just a collection of components, right, that work together. So a neural circuit is a collection of neurons that work together to accomplish tasks. So a simple circuit has components that go from the outside of the central nervous system through the central nervous system and then back out, right? So it goes peripheral, central, peripheral. And so one good example of such a simple circuit is a spinal reflex arc, right? So that just goes through the spine. And one good example of that is the myotatic stretch reflex, also known as the knee-jerk reflex, right? So you've all had the knee-jerk response before. When we talk about, oh, I've got this knee-jerk response to something, it kind of means that you have a reflexive response, right? And so that, of course, comes from the simple neurological reflex where if you hammer somebody's knee, right, then they kick their foot up a little bit. And so this is a prototypical example of a simple circuit that goes from a muscle which has some stretch receptors through an input into your spinal cord. Right? So this is a schematic of the direction in which the information goes. Right? So from the stretch receptors in the muscle to the spinal cord, which is of course part of the central nervous system, and then back out towards the muscles, which then elicit the movement, right? So actually execute the movement, right? So there's this one input, right, which is the sensory receptor, and then the axon, and then that gets converted into two outputs after having received the input into the central nervous system. Right, so there's a couple of really important terms here. One is afference, and another is efference. Right, so afferent, so anything that starts with an A means towards the central nervous system, towards the middle of the body. Whereas efferent, right, E for exit, it means exiting out of the body. So afferent inputs are inputs that go towards your central nervous system, right, from the periphery, and efferents are outputs that go from your central nervous system out towards the periphery. Another important couple of terms, dorsal and ventral. Right? So dorsal just means towards the back, whereas ventral means towards the belly. Right? And we'll come back and hammer this home a little bit more throughout the course of today. So what that input or afferent axon does, right? It takes information from the muscle sensory receptors through this input, through the dorsal root of the spinal cord, and this then gets converted into two outputs. So again, one input, two outputs. The first output goes directly from the afferent axon out through the spinal cord through the ventral root of the spinal cord towards the same muscle from which the input was received. So that's the first output. Now the second output, right, goes from the dorsal root of the spinal cord through an interneuron that then flips the signal, flips the potential, electrical potential of the input and converts that into the opposite response, right? Converts that into another output that again goes through the ventral root of the spinal cord towards the flexor muscle. Right? And so it's this interplay between the extensor and the flexor muscle that ultimately leads you to kick your foot. Right? And so that's again a one input and two output system the input goes from the receptor into the spinal cord. The two outputs, one goes back through the ventral horn of the spinal cord towards the same extensor muscle, whereas the other output goes towards the flexor muscle. Now, when you record in this neural circuit, right, you could actually stick electrodes into the different way stations of this simple pathway and actually record the signals, right? So of the action potentials as they're coming through, right? So you can see how when the sensory neuron comes in, right, there's an initial firing of the action potential, which has this all or none characteristic, right? So it starts negative and then it goes rapidly positive and then back down to even more negative, right? And then goes back into its resting state, right? So that's the input of the sensory neuron. And then there's a motor neuron, right? The first motor neuron actually goes, receives the same input and then immediately becomes more excited, right? So it becomes a little bit more likely to fire. And then in contrast, there's this mediating interneuron, right? That's in the middle of the circuit that flips the polarity of this excitatory postsynaptic potential towards an inhibitory postsynaptic potential, right? So that's how the two outputs are actually flipped relative to each other in their electrical signals, right? And so again, it's this interplay between the motor neurons that are in the flexor and extensor muscles that gives rise to the ultimate movement. So this is the simplest kind of circuit, right? One input, two outputs, a combination of excitatory input and excitatory postsynaptic and inhibitory postsynaptic potentials with interneurons that are mediating that circuit. Now, most cortical neurons, right, most neurons that are actually in your cortex are doing something in between, right? They're what we would call association neurons. So they include interneurons and a whole lot of neurons that are not just taking one input and converting that into one output, but are integrating many, many different inputs and then summing them towards their responses. But everything from the simplest to the most complex sensory and cognitive, as well as motor events, are enabled by these basic principles. So that's why I thought it'd be nice to start with a relatively simple circuit so that you can appreciate the exact inputs and outputs that happen, and then we can go on and talk, build on this and talk about more complex systems that give rise to behavior. Now that we've introduced the idea of neural circuits, let's start from the top down and talk about more generally the divisions of the nervous system. Now, as we talked about in the last day, there was this idea from Descartes of mind-body dualism, right? The idea of the separation between the soul and mind and the body. And Descartes famously thought that this was his idea of the reflex arc, the Cartesian reflex arc, is that the body and the mind work together as a kind of input-output system, where the body is acting as the inputs and the outputs, and the mind is a central level control system. And in some ways, that's actually been held true over the centuries in how we think about the organization, the fundamental organization or overview of the nervous system, right? So we think about how we have some sensors that are detecting the internal and external environment, right? External environment, things like sights and sounds and smells, right? And that then these stimuli from the external environment interact with sensory receptors, such as your skin and your retina and your cochlea and so on. Whereas internal stimuli, right, such as things like heart rate changes and other internal cues, interact with sensory ganglia and nerves and other parts of your peripheral nervous system, which we're going to talk about in a moment. So these are the sensory components of the overall nervous system, which then interact with the central nervous system, closest to what Descartes would have thought of as the soul, right? Where the mind actually is. Now the central nervous system, and we'll talk much more about its organization, but in general what it's doing is analyzing and integrating these sensory and motor information and then making some sense out of it, right? So acting as a central decision-making or executive process. Now that then goes towards output components, right? So in contrast to sensory components, which are inputs, you have output components, which are motor components, which are outputs, and that includes somatic and visceral motor systems, which then through motor nerves and autonomic ganglia and nerves, then go on to affect the effectors, which are the muscles themselves, right? So again, we have this general input-output organization with the central nervous system kind of acting as the high level, the top of the nervous system at a high level. Now, here is a more anatomical diagram of basically the same idea, right? But now it becomes quite clear how we have a central nervous system in contrast to the peripheral nervous system. Now, when people are talking about central nervous system, they just mean the brain and the spinal cord, right? So, the brain, obviously, right there. Spinal cord just goes down your spinal column, from the level of the brainstem all the way down your spine. Now, coming out of the spinal cord, you have the spinal nerves and the autonomic nervous system, and that's generally known as the peripheral nervous system, right? So the job of the peripheral nervous system, right, or PNS, is to interact or interface with these effectors, right? So such as your muscles and your cardiac muscles and skeletal muscles and so on, right? So actually to interface with these organs that then provide internal sensations and also external sensations and also motor systems. So this interplay between the CNS or central nervous system and the PNS and the peripheral nervous system basically acts out this input-output kind of process. Now, so if we zoom in on the central nervous system, right, you can see certain key structures of the CNS, and that includes gray matter and white matter. And gray matter generally consists of the cell bodies of neurons, right? And whereas white matter, in contrast, consists of the myelinated axons of the neurons. And that also includes a lot of glial cells and helper cells, and but importantly, the white matter enables the synaptic transmission of between neurons, right? So that whereas the neurons themselves are actually doing the computations that enable the action potentials themselves. Now, so what's interesting about the structure of the gray matter versus white matter, in general, when you're talking about the level of the brain, right? So here's a nice cross section of the brain, right? You can see how the gray matter generally is on the surface of the cortex, right, or the outline, right, the outside surface of the brain structure, right. So most of the outside of the brain, or the cortical surface, is full of gray matter, or the cell bodies themselves. Now, in contrast, the middle of the brain is richer in white matter. So there's more white matter, which again includes the axon bundles that enable connections between different neurons. Now, so that's quite well and good, but what's interesting is this pattern reverses when you get to the level of the brainstem and the spinal cord, right? So by the time you get to the spinal cord, right here is a cross-section of the spinal cord, and you can see how the pattern of gray matter and white matter reverses, such that at the level of the spinal cord, you have gray matter in the middle, towards the middle of the spinal cord, and white matter towards the outside, right? So the dorsal and ventral horn of the spinal cord, which we talked about earlier, that's where the connections actually go through these. And those tend to be gray matter-rich areas of the spinal cord. So there's this reversal in pattern, right, between outside and inside, and gray matter and white matter, where at the level of the spinal cord, gray matter tends to be in the middle, white matter tends to be on the outside, whereas at the level of the brain, white matter tends to be in the middle, and gray matter tends to be on the outside. And that has functional implications due to these structural differences. Okay, so when you talk about another way to kind of divide or subdivide this human central nervous system is at the level of different levels of the human central nervous system. And so we already talked about the spinal cord, and above the spinal cord we have the brainstem, and that includes the midbrain, pons, and medulla. And we'll talk a little bit more about specific functions of the brainstem in a moment. And then another major area is the cerebellum, right, which is so named as the smaller part. It's like the smaller part of the brain, right? It's like a smaller component of the cerebrum, which is the larger main, so-called main part of the brain. And then above the level of the brainstem and cerebellum, you have the forebrain, which it really includes the cerebrum. So that's the largest part by a long shot in humans, but that's not necessarily conserved across different species. And then we also have the diencephalon, which really interfaces between the brainstem and midbrain level and the cerebrum. Right. So that's the high level, top level of the human central nervous system, and then this, of course, then connects to the spinal cord, which runs down the length of your spine. And then the cervical nerves that are coming out of it interface with the peripheral nervous system. Now, so let's just zoom in a little bit in this middle part. Look at the green and red parts of this diagram. When you zoom in on that, you're looking at the human brainstem. Now here's the human brainstem. So this is the view from the front, or also known as ventral view. We'll go much more into this nomenclature in a moment. And this is the view from the back, also known as dorsal view. And so again, we can see the midbrain, pons, and medulla. You don't need to memorize every single word in these atlas type pictures, but what you do need to know is a little bit about the function of this of the human brainstem in general, and that's what's important to know is that the human brainstem is crucial for maintaining heart rate and blood pressure and breathing rate, basically your homeostatic functions, right? So the functions that enable you to stay alive. It also regulates the level of consciousness, right? So for instance, if you get a lesion in your midbrain, you're quite likely to be in a coma, right? So you'll have lost consciousness. And what's also interesting is that some of these areas actually also regulate some basic aspects of reward and pleasure. So in particular, there's this region within the midbrain that you can, that's visible from the dorsal view in an MRI, for instance, that's known as the ventral tegmental area, or VTA, and that's really important for coding for reward and pleasure. So that's one of the reward centers of the brain. Now, speaking about the brainstem, I've kind of mentioned a little bit that it's not conserved between different animals, different species. And what's quite interesting is that the brainstem is proportionally larger in area and volume in non-human animals. So this is a human brain versus a mouse brain. It's obviously not to scale. Mouse brain is obviously much smaller than the human brain. But when you compare, proportionally speaking, you could see how the human brain has much more cerebrum, right? So all that's in green here is the cerebrum, whereas the mouse brain has a proportionally much smaller cerebrum, cerebral cortex. Now, in contrast, the blue part, which includes the midbrain and the brainstem, are relatively larger in the mouse, right? And so this may suggest that these regulatory functions, homeostatic functions, and reward-driven circuits are relatively important for the mouse's survival, right? So there might be evolutionary reasons for this. Now here's just another example of this amazing difference in scale, a difference in proportional size in the brainstem between humans and other animals. So this is this is a two-scale comparison. A and C are two-scale comparisons of human brain and a trout brain, so a fish brain. And so the dark gray represents the cerebral cortex, the cerebrum, and so you can see how when you blow up the rainbow trout brain, the rainbow trout has proportionally much smaller cerebrum. However, the fish brain has a proportionally much larger brainstem, right? So again, these homeostatic functions are probably much more important, relatively speaking, to the trout. And so what's also interesting here is that you can see the conservation between the different additional areas of the brain. So right here we have also in blue, we have an olfactory bulb here. This is the human's olfactory bulb. We'll talk much more about this when we talk about the chemical senses. And so without this olfactory bulb, you can't smell. Now, relatively speaking, the mouse has a gigantic olfactory bulb, right? So, and the same with the trout, has a relatively large olfactory bulb relative to humans. So it might suggest that the olfactory sensations are proportionally more important for these non-human animals. All right, so you should be able to identify, from these general brain images, or diagrams of the brain rather, olfactory bulb, as well as forebrain, which includes the cerebellum, and then in contrast to the brainstem, right? And the brainstem, again, is proportionally larger in the non-human animals. Now, of course, having said so much about the brainstem, I should remind us all that most higher cognitive functions require more than the brainstem, or else you'd all be going brainstem storming. Since the brain is where it's at, of course, the brain is full of a few things: gray matter, white matter, and ventricles. And it's supported by some vasculature or arteries that provide blood supply towards the brain. So let's move on to talk about each of these major divisions of the brain structure itself, starting with gray matter structure. Now, and before we launch into all the gray matter areas that are in the brain, right? And I can just go on and on rattling all these different gray matter areas and what they do. But the first question, just to get things straight, is how do we get around in the brain, right? How do we navigate? So do we have some kind of map of the brain? Well, we do. We have lots of maps of the brain. But more modern neuroscientists are moving, not just using map, I guess moving from the old school method of using maps and compasses to point out places in the brain to much more modern techniques, such as more like a GPS system, right? So instead of saying, well, we're talking about an area dorsal of the temporal lobe, right, which is sort of a map and compass sort of view, a modern cognitive neuroscientist might say, well, we're talking about x equals 44, y equals 28, and z equals -2. So that's much more of a precise, I think of it as a GPS-based, coordinates-based system of navigating in the brain. But first, let's talk about the compass view. So in general, when you're talking about the major directions of the brain, right, we're not talking about north, south, east, and west. We're talking about superior, inferior, anterior, and posterior. Right, superior is anything that's above, inferior is anything that's below, anterior is in front of, and posterior is behind. Right, so if you say I'm looking for, I see activity in the brain in a region that's superior to the corpus callosum, right, then you'd have to find the corpus callosum and then find an area that's immediately above it, right? So it's a pretty useful relative term, a set of terms. Now, another useful relative set of terms is dorsal versus ventral, right? And we've mentioned this a little bit earlier today, but dorsal, it comes from the Latin word dor, D-O-R-S, which means back, and ventral comes from the Latin word ventre, which means stomach, right, or belly, right? So dorsal is towards the back, ventral is towards the belly. And of course, the reason why we're calling this the back is because humans have actually are one of the few animals that have evolved to stand upright on 2 feet, right? But most animals, four-legged animals, kind of walk around with their back to the sky, right? So more so in that sense, the dorsal direction would be aligned with the superior dimension, right? So in this pig, for instance, the more dorsal, the closer to the back, the more superior, right? Or the more above you're talking about. And then in contrast, the ventral surface is on the bottom or towards the side of the belly. Now, and so this anterior and posterior ends up being a little bit different, for a four-legged animal compared to in the human brain. Right? And so another term to know is rostral versus caudal, and that just follows the axis that goes, straight from the level of the spinal cord all the way to the very front of the brain. Right? And so rostral is towards the front of the brain, and caudal is towards the back of the brain. And then there are more ways to slice and dice this three-dimensional surface, or a three-dimensional volume that is the human brain and human body, and the animal other brain and body of other animals. But the dorsal versus ventral surface is the very important one to know. Dorsal, again, is the view from the back. Ventral is the view from the belly. Now, let's talk more about how we slice and dice the different views of the brain, right? And so these three terms are also quite important to know. One is coronal, and so that's just the frontal view, or this slicing of the brain in this direction. A sagittal view is the slicing of the brain down the middle, and axial is the slicing of the brain horizontally, right? So the way to remember that is coronal means crown. So if you can think of those, imagine those medieval paintings where they have, Jesus with a crown around his head, right? And it's like that's a corona around his head, and so it's just the lit up ring, that goes around the head. And that's a corona, right? So that way of viewing the head is the coronal view. Now, so this is a real MRI, and you can see quite clearly what I mean by sagittal, axial, and coronal sections, right? Sagittal section is the view from the side, right? So you can take many different slices, right, of this three-dimensional volume that is the brain MRI, and the slice right down the middle, which is this one right here, is called the mid-sagittal plane. Right? So this is not the mid-sagittal plane right here. It's just one of many possible sagittal planes. Okay. In contrast, the axial view is always the horizontal view, right? So you take a horizontal cut across, a horizontal plane across the brain, you're looking now at an axial view. And in contrast, the coronal view, again, is like a crown-like section, right? So if you take the brain, if you take a slice down the side, right, in this direction, then you're looking at a coronal view, right? And the reason why we have these different views, of course, is that these enable you to look at different specific structures of the brain, right? So, for instance, the hippocampus, which is a very important area for learning and memory in the brain, right? That tends to be more visible in the coronal sections and the sagittal sections, and not so visible in the axial sections, whereas the corpus callosum, which is the connection between left and right hemispheres of the brain, right, might be more visible in the axial and less in the coronal, and so on, right? So different sections enable different views of different areas of the brain. So that's why we need all three. Now let's go into the four major divisions of the human brain, right? And then we're going to apply how we think about these relative terms like dorsal and ventral, and sagittal and axial and coronal sections, and we're going to apply those terms towards the general ways in which we talk about the brain. So you've probably seen some kind of picture of brain like this before, right? It's probably in different textbooks even before you started this course. But right now we're looking at the lateral surface of the brain, right? Which means that we're just looking at it from the side, right, from the outside, from the side. So of course we have 4 major lobes, right? We have the temporal lobe, which is closest to your temporal bone, right, your temples. And then frontal lobe is in the front, and then parietal lobe is immediately behind the frontal lobe, and these are separated by the central sulcus, which is the central line or a fold down the middle or the center of the brain. And then in the posterior area part of the brain, right, so towards the back, you have the occipital lobe, which is important for vision and some higher order visual processes. So that's the lateral surface of the brain, where you can see all the major lobes. And then in contrast, you could have have a mesial surface of the brain, right? So that just means that you're viewing it from the middle. And so mesial just means from the middle, lateral just means from the side. Mesial surface, here you can still see the temporal, the frontal lobe and parietal lobe and occipital lobe, but you can see a little bit more of the occipital lobe, relatively speaking, but you can see a little bit less of the temporal lobe, relatively speaking, right? But what you can see now is some additional areas, and this here, I will talk about it more in a bit, but this is the corpus callosum, which is important, a really important bundle of white matter that connects the left and right hemispheres. You can also get a much better view of the brainstem and the midbrain areas from this mesial surface compared to from the lateral surface, and you can also get a much better view of the cerebellum. Now, so I've mentioned that the central sulcus here is a deep fold, right, within the brain that goes right down the center. So that leads us to another really important set of terminologies, which is sulci and gyri. Right? The sulcus is the singular, sulci is plural. Sulcus refer to the inward folds of the brain, and gyrus or gyri, plural, refer to the outward folds of the brain. Right? So sulci and gyri are the innies and outies of the brain. Now, and so that leads us to name different major areas according to whether they are inside or outside folds, right? So immediately anterior to the central sulcus, you have a precentral gyrus, right? So that's just a gyrus or an outward fold that's immediately right, or immediately before the central sulcus, right? So the precentral gyrus is immediately anterior to the central sulcus. And then, of course, the post-central gyrus, in contrast, is the outward fold, or gyrus, that is immediately after or post the central sulcus. All right, so you can talk about each of the major sulci and gyri of the brain this way. So the sulcus that divides the parietal lobe, right, which we've already talked about, and the occipital lobe, and that's known as the parietal occipital sulcus, right? Because that's the inward fold that divides the parietal and occipital lobe. So you can also apply specific terms towards each of the major gyri and sulci on the lateral surface of the brain. This looks like a pretty busy picture, but if you just think about how these terms are applied relative to each other, so here is the temporal lobe, right? And within the temporal lobe, there are some outward folds and some inward folds, right? And then there are some areas that are superior and some areas that are inferior. So the superior temporal gyrus, then, is just a gyrus that's at the top, right, superior part of the temporal lobe, right? So that's why it's called the superior temporal gyrus, right? And then immediately inferior to that, you have the middle temporal gyrus, which is in the middle part of the temporal lobe, and it's a gyrus, which is an outward fold. And then inferior to that, you have the inferior temporal gyrus. So this superior, middle, inferior temporal gyri are really quite logically named once you recognize the general terms. Right, so, and then we also have in the frontal lobe, right, the inferior frontal gyrus, which is just in the inferior part of the frontal lobe, and it's an outward fold, so it's a gyrus. So that's why it's an inferior frontal gyrus. And immediately superior to that, we have middle and superior frontal gyri. Right, so it's all quite logically named. I'm not going to give you on a test, one of these blind labeling diagrams to label, but you do need to know, generally speaking, what these terms mean, right? So think of it as an organizing principle with which you can navigate the brain space. So another way of thinking about the cerebral cortex, the brain in general, is by the different layers that it has. Once you zoom in on the gray matter structure of the cerebral cortex, you can see that they actually have different layers. So most neocortex, or relatively new, evolutionarily newer areas of the brain, such as the motor cortex and the visual cortex, they tend to have 6 layers. Right, the distribution is a little bit different between different parts of the cortex, but in general they have 6 layers. But there are some relatively older, evolutionarily older parts of the brain that have fewer levels, fewer layers. So for instance, the archicortex includes the hippocampus, which is important for learning and memory, and we'll talk much more about this later in this course, but for instance, it has only 4 layers, whereas the paleocortex, or piriform cortex, which includes the olfactory cortex. So we talked a little bit about the olfactory bulb. That then leads to the olfactory cortex, which is close to the front. It's in the frontal lobe. That paleocortex only has 3 layers, right? So again, while most parts of the cerebral cortex have 6 layers, some relatively older areas of the cerebral cortex tend to have fewer. Another way to slice and dice the organization of the cerebral cortex is by numbering systems that previous scientists have devised that divides the cerebral cortex into a certain number of areas. So Brodmann was famously a German neurologist who very carefully did cytoarchitectonic work, right? So he did microscopy to use the different general structures of neighboring pieces of cortex to make this parcellation or to define different areas of the cerebral cortex. And so he came up with 50 different cortical areas that he divided the brain into, right? So and so this is just one of many maps that are out there in the in neuroscience right now, with which you can divide up the general human brain, right? And this is just useful as a way to communicate with other scientists, where exactly you're talking about in the brain when you, for instance, see activity in a certain area. You'll say, oh, I saw activity in area 44 and 45, right? So if you tell somebody that, then a knowledgeable neuroscientist would be able to know that they're talking about the inferior portion of the frontal lobe. So that means the inferior frontal gyrus, right? So that's areas 44 and 45. So we've talked quite a bit about gray matter structure and anatomy. Let's move on to talk about the ventricles, right, and the fluid-filled space, which are the fluid-filled spaces, and the vasculature, which are the arteries of the blood supply to the brain, before we move on to talk about white matter structure. Now, so the ventricular system, there's not so much in your textbook about ventricular system, but I do think it's quite important to know that the brain isn't just all a lump of gray and white matter. It's not just all cells, but there are spaces between these cells as well. And so a lot of those spaces are linked together in a major ventricular system. Ventricles just means spaces. And what's important to know is that these ventricles are interconnected, and so they're named different things. You don't really have to know about the nomenclature of those individual ventricles, just need to know that they go from 1 to 4, and there's lateral ventricles, which are on the lateral, so that's closer to the side, of course, but then they're connected via the 3rd and the 4th ventricle, and then all the way down through to the spinal cord, right? So this ventricular system is a interconnected set of fluid-filled structures that are filled with cerebrospinal fluid, or CSF, and they go all the way down your central nervous system, down your spinal cord. Now, this is another view of the ventricular system. Again, we've got the lateral ventricles, right, that are then connected to the 3rd ventricle and the 4th ventricle, and then through the central canal all the way down the spinal cord. Here's just another view of basically the same thing, but I thought this was a very nice demonstration of how how interconnected those ventricles are. You can actually develop a 3D model of the whole system as one individual interconnected series. Now, and the reason why I keep pointing this out is that you can get a lot of information from the status of the brain by taking samples of the cerebrospinal fluid through the central canal down your spinal cord. And so this will be interesting for some of you who might be pre-med. If you ever get asked to do a lumbar puncture, I don't know how many of you have had a lumbar puncture. I wouldn't wish it on anyone. This happened to me when I was, a few years ago. I got the worst headache of my life, probably just because I didn't drink enough water. And having been in a neurology department for long enough, I thought, well, worst headache of your life, you could be getting a stroke. So this could be really damaging. So you better go see a doctor real quick. So I dialed 911 and went to the emergency room, and then they said, well, you have the worst headache of your life. Well, it's probably nothing, but just to make sure that you're not about to have a stroke, or not having a stroke, we're going to do a lumbar puncture. And what that means is they make a hole in your spinal column, and they draw from the spinal column a small sample of your cerebrospinal fluid. Now, and then they analyze the cerebrospinal fluid, and if there are red blood cells, right, if there's some blood in the CSF, then that means that you might have internal hemorrhaging or stroke or other kinds of internal bleeding in the brain, right? So the cerebrospinal fluid is very important for giving a good clinical indicator of the status of a patient. So that's a little bit about the ventricular system. Let's talk also about the blood supply to the brain. So this is just a model of blood supplies, and you can see that it's quite a tangled mess. Now, but what I do want to point out is that we have some larger blood vessels that are connected to smaller blood vessels, right? So the larger ones are arteries that then go towards the capillaries, which are smaller blood vessels, that all of which innervate the brain. Now, so this is another view of the blood supply to the brain. What's quite important when we're talking about brain blood supply, to know is that the blood comes from through the arteries, right, from internal carotid arteries all the way up to the brain, and at the bottom of the brain we have this important structure known as the circle of Willis. And that the circle of Willis is kind of the basis of the blood supply towards the rest of the brain. So this is a ventral view of the brain now, and you can see how they've cut out they've drawn this portion of the temporal lobe removed so that you can see how the different arteries come out of the circle of Willis. So circle of Willis is this area right here, and then from the circle of Willis you have the different major cerebral arteries that then go out towards the different parts of the brain. Now, so this is yet another view of the circle of Willis, but now you can see quite clearly coming out of the circle of Willis. Oh, this is Reverend Thomas Willis, by the way, after whom the circle of Willis is named. But coming out of the circle of Willis, you have a few major arteries. And so these are important to know because they have clinical significance as well as just important anatomical landmarks. So we have these cerebral arteries. So there are three major cerebral arteries, and those are the anterior cerebral artery, the middle cerebral artery, and the posterior cerebral artery. Right, so they provide blood supply to the front of the brain, anterior, the middle of the brain, the middle cerebral artery, and the posterior, the back of the brain, right, is the posterior cerebral artery. And so, and in between these major cerebral arteries, we also have communicating arteries, right? So the posterior communicating artery is just the artery that communicates towards the posterior cerebral artery. And so this is another view of the blood supply to the brain. And so while the previous couple of images were ventral views. This one is now lateral and mesial view of the brain, right? So this is already becoming very important. Now, what's shown here is the differences in blood supply towards the anterior part of the brain, which is mostly supplied by the anterior cerebral artery, and the middle part of the brain, which is mostly supplied by the middle cerebral artery, and the posterior part of the brain, which is supplied, of course, by the posterior cerebral artery. And what's quite interesting is you can start to see, right? You can appreciate how different parts of the brain are supplied by different arteries. But what's also of clinical importance here is that you can start to see some territories that tend to be supplied by the different arteries, and then you can start to see the boundaries between those different territories, right? So we're color coding the areas of the brain that are supplied by the ACA and the MCA and the PCA, but the dividing areas are where you tend to have less blood supply, right? And you tend to have less blood pressure towards those areas, because those are areas that are kind of in the watershed, right? They're kind of in the dividing zone between ACA and MCA and PCA, right? So these watershed areas are the overlap between the distal ends, or the far ends of two arteries, right? So you can have the watershed area between the ACA and the MCA. You can have the watershed area between MCA and PCA. And what's important there clinically is that you might, in some cases, have not enough blood pressure or hypotension towards these arteries, and that puts these watershed areas in a danger zone for infarcts or tissue death due to the lack of oxygen in the area. Right. So obviously, the purpose of having blood supply is to provide oxygen to areas of throughout the brain, and in these areas where these watershed areas where you might have not enough blood pressure, you are at a greater risk of stroke in those areas. So, and watershed infarct, it's just is cell death as a result of limited oxygen towards these watershed areas. Now let's move on to talk about the last section of what I'll cover today, which is about white matter structure and anatomy. Now, we've already covered how the brain includes gray versus white matter. Right? Gray matter includes the cell bodies of the neurons, whereas white matter includes primarily the axons and the myelinated bundles that are wrapped by Schwann cells containing myelin around those axons. Right. So while gray matter are the cell bodies themselves, white matter tend to be the connections between the cell bodies. Now, so when you have large collections of cell bodies, that they are then connected to large collections of myelinated axons, which together are known as tracts. Right. So a tract is just a large collection of myelinated axons. And then when you have a large bundle of tracts all together, you have what's called a fasciculus. Right? So a fasciculus, or plural fasciculi, is just a bundle of white matter tracts. And these bundles of tracts tend to travel in coherent directions. Right? So you can have ascending fibers versus descending fibers. Right? Ascending fibers tend to be sensory fibers. Descending fibers tend to be motor fibers. But what you can see here, this is just a schematic of a bundle of fibers and tracts that are traveling in the same direction, right? Ascending or descending. Now, so fibers and tracts and fasciculi, they run in coherent directions, and then and they actually also start to form different a large collection altogether, and what you can describe them as are different kinds of fibers that are commissural fibers, or versus association fibers versus projection fibers. Right? So commissural fibers are the fibers that run left to right in the brain. Right? So between the different hemispheres. For instance, the corpus callosum, which is a major white matter pathway, white matter tract that runs between the left and right hemispheres, that's a commissural fiber. Now, in contrast, an association fiber is a fiber that runs in the anterior-posterior direction. So they tend to be within the same hemisphere. You can have an association fiber that goes front to back. Right. And then a third kind of fiber, right, the third direction, is projection fibers, which are shown in the Z direction here. And those are fibers that project superior to inferior, or they run top to bottom in the brain. Neuroscientists, specifically researchers and clinicians that use neuroimaging, have come up with a convention for color coding these different fibers. So in general, area of fibers that run left to right, which are commissural fibers, tend to be labeled red, and in different brain imaging scans, which I'll talk much more about in the next lecture. Whereas association fibers tend to be color-coded left to right, whereas projection fibers tend to be coded, which run top to bottom, tend to be coded blue. Right, so again, you have association fibers, which are front to back, projection fibers, which are top to bottom, and the commissural fibers, which are left to right. So when you put all the color codes together, you can get a major overarching view of the white matter of the brain, such as this one. And again, you can see that the computer algorithm has color coded each of these fibers in a different color depending on their major direction that it's traveling in. Right, so you can see how this bundle of green here, right, so green is coding for association fibers, which are running front to back, right? And so that's one major fasciculus, right, or a bundle of fibers, white matter tract in the brain. This blue here, right, that's a projection fiber, which is going top to bottom in the brain, whereas the red here, you don't see as much red just because of there's so much green and blue in the way, but the red fiber is color coding for fasciculus that is going left to right. So as a good example, a corpus callosum is composed of commissural fibers, right, because they're going from left to right. So this is another final view, right? So this is using a different color coding scheme now, but what the scientists did to enable this view of all the white matter pathways in the brain is they're taking each of the anatomical landmarks of gray matter and then finding from those anatomical landmarks major white matter bundles that go towards that area of the brain. So each of these white matter bundles is labeled with with its corresponding name. So the cortical spinal tract, for instance, is the white matter pathway that's going top to bottom. Superior longitudinal fasciculus is a pathway that connects longitudinally, so front to back, so it's an association pathway, an association fiber, and it's relatively superior in the brain, and it's a fasciculus, which is means a white matter bundle. So again, I'm not expecting you to memorize all these names of white matter pathways in the brain, but you do have to know that they're generally named after the direction in which they're aligned, and then also they're named after the gray matter endpoints that they connect. So I'll close by treating you with with this overview. I've done lots and lots of talking about different brain areas, but of course there's nothing more compelling than a song. So here is The Brain, sung by The Brain. Enjoy!
And now, the parts of the brain performed by the brain. frontal lobe, hippocampus, neural node, right hemisphere, pons and cortex, visual, Sylvian fissure, pineal, left hemisphere, cerebellum left, cerebellum right, synapse, hypothalamus, striatum, dendrite. Axon fibers, matter gray, central tegmental pathway, temporal lobe, white core matter, forebrain skull, central fissure, cord spinal, parietal, diameter, meningeal vein, medulla oblongata, and lobe limbic, microelectrodes, the brain. That ought to keep the little squirts happy. Yeah.